So OmicsLogic Transcriptomics is an online training program that is designed for students that do have the background in bioinformatics and also don't have this background in bioinformatics. So if you already have a background in bioinformatics, perhaps you are familiar with things like genomic data analysis, maybe processing multiple sequence alignment, or maybe working with structures. And all of these are very useful tools to start working with transcriptomic data. But there are specific things that you need to be prepared to do if you are trying to analyze the bulk RNA-Seq data or the single cell RNA-Seq data that is going to come with an additional set of challenges. And so that's really what we want to address today. For those of you that have no background in transcriptomics or in bioinformatics at all, we want to explain how, even though you don't know, you can still benefit from understanding the logic behind some of the analyses and see some examples that will relate to the biology that you might understand really well. So really to appreciate how RNA-Seq is used for real-world biomedical research applications, including basic and translational research. So in this program, we will see how relying on analytical methods and reference databases, researchers can study RNA-Seq data and understand how gene expression variation can really help us understand biology that is described by this RNA-Seq data across many conditions. And so to do so effectively, bioinformaticians leverage big data processing methods. So how can you actually take gigabytes and maybe in some cases terabytes of data and convert them to structured data, to tables of expression. Then we'll talk about how exploring the variability across this kind of structured data can help us find major trends and patterns. Then we'll talk about statistical analysis. So what are some of the commonly used statistical approaches to test hypotheses and find variation between groups of samples or across many different types of samples, so maybe multiple classes or multiple types of samples that we have. And finally, we'll talk about data mining and machine learning, which will introduce us to single cell RNA-Seq. And single cell RNA-Seq is a growing popular set of methods. They rely on kind of a lot of machine learning that we will introduce you in the context of bulk RNA sequencing, but there are also specific packages that we will talk about around single cell RNA-Seq. So why next generation sequencing, right? So when we speak about next generation sequencing, we're really trying to understand how proteins that are expressed could be measured using a proxy. So using mRNA as a proxy for protein expression. Why protein expression? Well, obviously protein expression is what defines the phenotype of the cell. We can understand a lot of things by measuring, for example, surface proteins. We can understand the phenotype. What do these cells actually do? How do they interact with cells around them? We can also start looking at the internal processes by measuring mRNA, which we cannot do if we are looking just at, for example, immunohistochemistry. And so many different methods have been designed to measure mature mRNA, including, for example, microarrays. And microarrays are a very good way to measure predefined levels of mRNA. But if we are looking at next generation sequencing, we're really getting a much higher dynamic range, which means that we can start looking deeper into alternative splicing, into different transcripts, into how we can actually find exons that might better give us a sense of separation between specific groups of samples, as you can see right here. So we can really start looking at all of the biological activity at the transcription level, and we can try to correlate that with phenotypes that we know to have both mechanistic significance, but also clinical significance in terms of biomarkers for diagnostics and maybe even for treatment selection. So what we have been doing with our training modules is we've been trying to design these kinds of training programs around multi-omics data sets. And that means that we are really focused on taking a comprehensive systems biology look at how biological processes that happen within the cell can be interpreted to apply them to diagnostic and treatment selection challenges for novel drug discovery, drug repurposing, as well as diagnostics using and leveraging large data sets. So that's what the concept of OmicsLogic really stands for. And in the context of this program, we're going to kind of take a narrow look at transcriptomic data, which is a very comprehensive type of data, especially in application to different types of projects. And so what kinds of things could be used, could RNA-Seq data be used for? So as I mentioned, cancer biology. So to understand this complex disease that we call cancer, we really need to understand how different processes inside cancer cells develop at the individual level. And so we will present you with a few case studies to really appreciate the variability across many different types of and subtypes of cancer. And then we will also take a look at how we can classify disease and stratify our patients. How can we actually understand individual patients in the context of what we call a diagnostic state? A diagnostic state is a specific diagnosis that a doctor would assign their patient to, and we will try and see how individual patient variability fits into that paradigm. We will also take a look at single cell and tissue composition. So we will actually take a look at how single cell data can reveal some important differences between specific types of tissues and cells, for example, immune cells, or different types of treatment related specificity of cell states. And we will also look at drug response, which is, of course, different across many different cell types. So how will we do this? In this program, we will cover 6 different topics. We will provide you with an overview of some standard tools and resources for transcriptomic data analysis. We will take a look at processing next-generation sequencing data using bioinformatics pipelines for pre-processing, mapping, and quantification. We will take a look at analysis, normalization, and visualization of gene and isoform expression. We will give you an introduction to big data mining, machine learning, and predictive analysis, and also introduce you to analysis of single-cell RNA-Seq data for tissue-specific expression, and finally introduce you to project examples. Now, the project examples, I think, are critical here because especially when you study a complex topic like RNA-Seq, it really comes to life when you have a very specific challenge that you would like to address. So that's really going to be the focus for what we do. Now, one of the tools that's going to be extremely helpful in this process is the T-BIOINFO multi-omics platform. Why is this important, both for bioinformaticians and for non-bioinformaticians? As a bioinformatician, you probably are familiar with the challenges in dealing with large data sets. So again, we're talking about gigabytes and even terabytes of data. Processing those on your computer just does not make sense. So you have to have access to infrastructure that can support this kind of analysis. And that's what this platform provides in the context of this program. So you'll be able to understand some of the principles using example pipelines, and then you will be able to try and apply some of these to a data set of your own. For those of you that do not have a background in bioinformatics, this is really a way to understand what the methods do without having to deal with some of the technical challenges. So you don't have to know Linux, you don't have to know bash scripting, you don't really need to know a lot of the settings that are really essential for processing RNA-Seq data effectively. And I want to show you how. So let's put a link here just for our participants to the platform, and I will give you a brief overview of what this will look like. So the T-BIOINFO platform combines multiple methods of analysis into logical workflows. These logical workflows are color-coded and linked to the logic behind the analysis itself. So you will see color coding in this RNA-Seq processing pipeline for pre-processing, mapping, and quantification. And as a result, you'll be able to compare many different variations of the same pipeline to appreciate the different approaches for pre-processing, for mapping on the genome, on the transcriptome, or even alignment-free quantification, and understand some of the differences between different types of normalization techniques and different approaches to actually prepare that table of expression to ultimately perform downstream analysis. And by downstream analysis, I mean standard type of analysis like differential gene expression, as well as mapping that on biological processes, as well as interpreting the meaning of a differential expression analysis result in the context of pathways, right? And so that's going to be one of the ultimate goals for this program, is for you to be able to go from the very beginning, where you take the raw data, for example, raw data that looks like this, with short reads that are mapped to a particular portion of the genome. You can then use the mapping of these short reads to apply to quantification of whole genes that could be thousands of nucleotides long. And then using that information to prepare a structured table of analysis, know how to explore that table of analysis, detect major patterns, find outliers, eliminate noise, and then go to a biologically meaningful visualization that you can start building your understanding on. To do that, we're actually going to look at many different things, like visualization of basic patterns, really to understand what do these genes look like, how do we compare them across conditions. We will talk about normalization and transformation, how we can prepare the data to fit some of the assumptions that we will rely on to deploy the statistical and analytical methods that we will use after that. For example, log scale transformation, and we will learn how to do that in both Excel, R, and Python. And then we will look at how we can find these meaningful patterns that we either want to find, so using supervised machine learning, or find new patterns that we yet have not discovered using data mining. Now, as you know, there are many challenges along the way. And when you face a real research project, what you want to know is how do these different methods work together, and how can they reveal important parts of your analysis to you, right? So how to actually get the thing that you need from this type of analysis. And an important part in that is performing exploratory analysis to find these significant biological signals. Now, to do that, what we will do is we will introduce you to the logic behind this, and then also offer you some asynchronous resources for training. So as I mentioned, a lot of this could be done on the platform. A lot of the downstream exploratory analysis and visualization, you can try to do it in Excel even, but the most practical way is to learn a little bit of coding at least, and even better if you become somewhat proficient in some of the coding approaches. So we will give you tutorials in R and Python. For example, in R, you will be able to perform the full downstream differential expression analysis with descriptive statistics, with volcano plots and MA plots, and then export tables of differentially expressed genes, and really know how to run this independently on your own computer, and obviously in an online environment that we will introduce you to. Now, what's important is that I think a lot of people confuse being familiar with some coding practices, and really knowing how to do RNA-Seq data analysis. So one of the things that we focus on is the logic, and the logic is kind of code agnostic. And so that's why we'll provide you with similar tutorials in both R and Python, how to perform all of the steps that are needed to get what you need from the data. We will do this first on the OmicsLogic portal, where you have predefined assignments for you to complete and really appreciate what kinds of outputs you are expecting to get. And then you will be able to download all of this code and work on this code independently on your own computer, so that you can get the types of outputs that you need. And you can really make sure that you are coming away from this training with a set of different codes that are ready for you to do analysis on a data set of your own. Now, what kinds of exercises do we provide on the portal? So on the Learn portal, inside of the Transcriptomics portal, the Transcriptomics program, you will find sessions, which will be interactive sessions like what we're having now. Then you will be directed to these asynchronous resources that you can read through and do some practical exercises, from basic analysis pipelines to exploratory and downstream analysis using R and Python, and all the way to biological interpretation. So this program will really provide you with a comprehensive set of resources, and the program sessions will link these resources into a meaningful flow of information and practice, really going from the very basics all the way up until the expert level. Now, as you know, single cell transcriptomics has become one of the hottest, most interesting areas of research. We're talking today about multi-omics single cell sequencing. We're talking about single cell proteomics and even metabolomics. So really, single cell has become a very mainstream method that a lot of labs are trying to apply to basic research questions, and even some to translational research projects as well. And so what you see on the slide is kind of a timeline for when different single cell protocols started to get published. And we go from all the way back in 2009, when the first manual protocol was designed for single cell, and all the way up until 2017, approximately 10x genomics has become kind of one of the mainstream methods today. And today we have an ever-growing number of protocols for different types of applications. And so what we will focus in this program is on transcriptomics using single cell data. How is single cell transcriptomics data different from bulk RNA-Seq data? The main difference is that instead of measuring the average temperature in a hospital, you're able to go to each individual patient and understand who has high fever and who has died, right, and has no temperature at all. And so if you take the average of those two, somebody with very high fever and somebody who's dead, you will end up with somebody who's not even sick, somebody who's maybe healthy. And so to understand the variability of patients in the hospital, you really want to go to each individual. And with single cell RNA-Seq data, it's the same thing. Now, because of that level of information, you also get a lot of noise, and you also get a lot of confusing signal. And so that's why we will take a look at very specific types of analysis. So we will look at methods like Monocle and seurat. We'll look at pseudotime analysis, at clustering, differential gene expression. We will take a look at in-depth seurat methods, where we will explore how to make visualization that is typically something that you will expect to find in a publication, for example, your UMAP plots. We will take a look at differential expression in the context of different marker genes. We will take a look at pseudotime and what are some of the considerations around preparing the data for pseudotime analysis. So all of these are going to be very helpful skills if you're going to try and deal with a single cell data set. Now, as I mentioned, all of this by itself is pure theory. And so what's important to apply this kind of information to a project is to give you practical hands-on sense of how do you go from raw data to biological interpretation. And that's why we have designed some use cases around project examples using cell line data, xenograft models, as well as clinical trials and some translational applications. So we will take examples from high throughput testing of drugs on cell lines. We'll take a look at tumor microenvironment in animal models, and we'll also look at TCGA data, the Cancer Genome Atlas data, to try and appreciate real patient data. What does that actually look like? Now, in terms of the types of things that we will get out from this is we will look at the evolution of breast cancer classification. One of the most significant progresses in diagnostics and therapeutics has been made in the area of breast cancer. And one of the reasons for that is because from just a very high-level diagnosis based on marker genes, we are able to now go into the deep molecular biology in each subtype of breast cancer and understand how it relates to treatment. And to do that, we'll take a look at actual publications. So we'll take publications that have made a significant impact on this area, and we'll make sure that we can understand all of the specifics of the methods that the authors have used. And these are publications that have been produced by both researchers as well as people in the industry. So these are going to be papers from companies like Genentech, Novartis, and others that have really made the progress with actual therapeutics and diagnostic strategies for these diseases. Now, beyond that, we will take a look at patient-derived xenograft models, modeling cancer precision medicine, immune changes in macrophages, TCGA, and even we'll take a look at many other types of projects. So really a lot of projects. I'm not going to go into all of them, but especially I want to kind of highlight how this could be relevant to some of you that might be either doing your master's or maybe PhDs in different areas. So we have here an example of Olga Udartseva. She's a research affiliate, a postdoctoral research affiliate at Roswell Park Comprehensive Cancer Center. And this is what she has gotten out of a program like this. She says the program is very well structured and easy to learn. The skills that I gained through the program helped me significantly enhance the relevance of my project by making connections between my preclinical study and its practical applications to cancer patients. So really, I hope that this would be an outcome for all of you that decide to join this training program. So to speak more about the different